what is the benefit of glycolysis? Why glycolysis happens inside the cell? The basic idea of glycolysis inside the cell is the conversion of glucose, as I said, is to form pyruvate. But it gives a bigger picture to form pyruvate, and in the process through substrate level phosphorylation some ATP is formed, and also some electron donors like NADH is formed, and this ATP is used as energy inside the cell because ATP is the energy currency of the cell, whereas the NADH is used in the electron transport chain, the ETC, inside the mitochondria to generate more ATP, because this electron transport chain pumps out protons, as I have discussed in the video that is listed here. So it pumps out protons inside the mitochondrial membrane, and when the protons enter back through the ATP synthase enzyme, it produces ATP through the process of oxidative phosphorylation. So this is basically the product of glycolysis is the reactant, the NADH is the reactant of ETC, and the ATP produced is used as it is, and the pyruvate that is formed by glycolysis gets converted to acetyl-CoA, which is a 2-carbon sugar inside the mitochondria, and this acetyl-CoA enters the Krebs cycle, and through this Krebs cycle what happens is that more of ATP, more of NADH, FADH2, these are produced in the cycle, and they are fed back into the ETC, which again leads to the formation of ATP. So the final goal of all this, the glycolysis, the Krebs cycle, is the production of ATP, and glycolysis is the initiator. It initiates the breakdown of sugar. So the sugar that is used by the cells, that is the glucose, leads to formation of pyruvate inside the cell, which acts as the reactant for the Krebs cycle. It starts the process, it kickstarts the process. The glycolysis has the goal to form pyruvate, which can move to the mitochondria, because glucose itself cannot move inside the mitochondria because it is a very large molecule. So it has to be converted into a 3-carbon molecule so it can move inside mitochondria, and everything else can take place. Glycolysis has 10 steps. In the first 5 steps, the 1 to 5th step, energy is required by the cycle, and this energy is provided by ATP. In the 6th to 10th step, energy is produced inside the cell. I have discussed this in the starting of the video also, and the energy is also in the form of ATP. And the 1st to 5th step are called as the prep phase or the preparatory phase, whereas the 6th to 10th is called as the payoff phase, because it pays off in the form of energy, and also electron donors are formed. And what happens is that in the preparatory phase, the cell has to break down glucose. So in the preparatory phase, in the prep phase, the C6 or the glucose breaks down to the C3, okay, and in the payoff phase, where we get ATP, where we get NADH, in the payoff, what happens is that the C3 changes forms from glyceraldehyde 3-phosphate to pyruvate, but in the process it releases ATP and NADH. Okay, in the payoff phase, the C3 changes to C3 only, so no loss of carbon takes place, but in the process ATP and NADH are produced. So basically the lysis, the breakdown happens in the preparatory phase, whereas the result, the outcome, the benefit of that lysis we receive in the payoff phase. Glycolysis does not run without checkpoints. It requires checkpoints in various places for the cycle to run properly. Okay, I will discuss all the checkpoints. There are three major checkpoints that are there in glycolysis where the cycle has to overcome those checkpoints to move forward. And the glycolysis happens inside the cell when the concentration of glucose in the cell is high. So glucose concentration is high, ADP concentration is high, which means that the concentration of ATP is low, and after glycolysis the concentration of ATP is high, and also the proton donors, the NADH is lower in concentration. So if NADH is lower in concentration, which means that the NAD positive is higher in concentration inside the cell. These are the factors that run glycolysis, and the lower concentration of ATP and NADH also makes the glycolysis move in forward direction. But the inhibitors of glycolysis, which inhibit the reaction, these are the inhibitors also. If I talk about the inhibitors which inhibit the reaction, inhibitors are if ATP concentration is high inside the cell, then it acts as an inhibitor. If NADH concentration is high, it acts as an inhibitor. If glucose concentration is low, it acts as an inhibitor for the glycolysis. So glycolysis in each and every phase needs some reactants to form product, and the concentration of reactants determines if the glycolysis cycle will move forward or not. And the products that are formed in different stages and different steps of glycolysis are used by other biochemical cycles inside the cell, such as they are used in gluconeogenesis, protein synthesis, amino acid synthesis, fatty acid synthesis, and so on. So in different cycles they are used. So now I will be talking about the different steps of glycolysis. The steps of glycolysis, the 10 steps in detail, and why they are necessary and why they happen in the sequence they are happening inside the cell. And for the legend I will be using the reactants with red color, the enzymes with green color for the particular step, and the product with blue color. Okay, so in the first step of glycolysis, the step number 1, in the first step of glycolysis, this glucose is converted to glucose 6-phosphate. So a phosphate group is added to glucose. So glucose gets converted glucose 6-phosphate by the action of enzyme hexokinase. So as it is the first 5 steps are the preparatory phase, so the cell has to invest in this phase. Glucose gets converted to glucose 6-phosphate by the enzyme hexokinase, and hexokinase is a kind of transferase. As I was discussing about the types of enzyme, I talked that wherever you encounter a word kinase, it means that the energy for the transfer of functional groups takes place by the breakdown of ATP. Wherever kinases are there, the breakdown of ATP takes place, or in the reverse reaction, when a high energy substrate with phosphate group is present, then the production of ATP takes place. But here we see that the glucose has been converted to glucose 6 phosphate, which means that the phosphate group has been transferred to glucose. So the glucose here is low energy, whereas the glucose 6 phosphate here is high energy, and the energy is being provided by the ATP. So in this process, a ATP molecule is converted to ADP. Okay. So this is the first phase of glycolysis where energy is used. The first cycle of glycolysis where energy is consumed in this process, which means one ATP molecule is used in the first phase, in the first step of glycolysis, and this step also acts as a checkpoint. So this is one of the checkpoint of glycolysis, the first checkpoint of glycolysis. Glucose is converted to glucose 6 phosphate is because the glucose can easily enter or exit the cell through transporters. So various glucose transporters are there on the cell membrane, and glucose can be easily transported inside and outside the cell through these transporter. But once glycolysis starts, the glucose has to be converted to glucose 6 phosphate. Adding a phosphate to glucose makes the glucose impermeable to the membrane. So glucose 6 phosphate, because it has a phosphate group which is negatively charged, it is repelled by the cell membrane, so it cannot exit the cell. So it is the first checkpoint of glycolysis where the glycolysis initiates itself. The glucose is converted by the action of hexokinase to glucose 6 phosphate, and doing so makes it impermeable to cross the cellular membrane. So now the glucose cannot cross the cell membrane and is retained inside the cell. So this process is necessary for the retention of glucose inside the cell. So that's why it is called as the first checkpoint, because now the glucose is bound to undergo glycolysis or is retained inside the cell, so it can be used in other cycles as well. Glucose 6 phosphate is a reactant for other cycles also, so it can go into the other cycles and can move forward to glycolysis. So in the next step of glycolysis, the glucose 6 phosphate gets converted to fructose 6 phosphate by the action of enzyme phosphoglucose isomerase. So by the action of enzyme phosphoglucose isomerase, it gets converted to fructose 6 phosphate. Phosphoglucose isomerase. So it is an isomerase enzyme, which means that it can only change the structure of the compound and not its chemical formula. So fructose is also C6H12O6 and it is linked to the phosphate. So glucose is also C6H12O6. So simply it converts the glucose 6 phosphate to fructose 6 phosphate through the action of phosphoglucose isomerase. It is an isomerase enzyme, changes the structure. So now it is easy to remember because it isomerase, so it changes structure and not changes formula. So not changes chemical formula. By the name of the enzyme you will easily remember it, and phosphoglucose means it's the name of the substrate which is acting upon it. So glucose phosphoglucose isomerase is acting upon glucose 6-phosphate. So the name of the substrate, it is an isomerase enzyme. It makes fructose 6-phosphate, and only the structure changes from an aldehydic sugar to a ketonic sugar. So glucose is aldehyde. So this one is aldehyde, and this one is a keto or a keto sugar. So it only converts the aldehydic sugar to a keto sugar. So in the third step of glycolysis, the fructose 6-phosphate gets converted into fructose 1,6-bisphosphate. Fructose 6-phosphate, it adds another phosphate to it and becomes fructose 1,6-bisphosphate. And the addition of phosphate in fructose 1,6-bisphosphate is through a kinase which is called as a phosphofructokinase. So here also a kinase comes in, it's called as phosphofructokinase. The names are easy to remember because it has a fructose 6-phosphate as a substrate, so phosphofructokinase. And this is also a transferase, and the kinase name kicks in, which means that the phosphate that is provided to the fructose comes from ATP. So here also the ATP provides the phosphate for the formation of fructose 1,6-bisphosphate. So one more ATP is consumed in the preparatory phase of the glycolysis in the third step. And this step adds the phosphate to the first and last carbon. So phosphate is added to terminal carbons of molecule. So the terminal carbons have the phosphate, means they are charged up, they are full of energy because they are bounded by high energy phosphate bond, and the terminal carbons have phosphate group on it. And this is also through the enzyme phosphofructokinase, and it is the second checkpoint of glycolysis. Why it is the second checkpoint? Because as you all know, in the first checkpoint the glucose formed glucose 6-phosphate because it allowed the glucose 6-phosphate to remain inside the cell and not to move out. So it retained the glucose inside the cell, and in this form, the fructose 1,6-bisphosphate, it charges up the fructose for breakdown in the next step of glycolysis. So in the next step of glycolysis, the 6-carbon sugar, which is being charged up by 2 phosphates, okay, because it has to break down into 2 3-carbon sugars. So both sugars require their respective phosphate groups. That's why this has terminal phosphate groups charged. So the C6, the fructose that is charged with phosphates, is breaking down in the next step. So it is making the sugar ready. So in the next step, breakdown happens and leads to the formation of 2C3 carbon, which is the end of preparatory phase of glycolysis. So after the sugars break down into 3-carbon sugars, the preparatory phase is over. This step also means that now the sugar, the C6 sugar, has been primed up for breakdown, and this step is irreversible now. So moving forward, glycolysis will happen, it will not subside, it will not end. Glycolysis has to happen. So glycolysis from this point irreversible. Okay, The glycolysis cannot move in the backward direction, it cannot subside. Now we come to the fourth step of glycolysis. In the fourth step, it is very simple, as I have discussed in detail in the last step. Now the fructose 1,6 bisphosphate has to undergo breakdown. Bisphosphate has to undergo breakdown, and the breakdown is caused by the enzyme aldolase, and aldolase is a lyase, and it forms one DHAP and one glyceraldehyde phosphate. So it forms two products because it is a C6. So fructose 1,6 bisphosphate is a 6 carbon sugar and forms 2,3 carbon sugar. So this is also C3 and this is also C3. So it forms 2,3 carbon sugars, and one sugar is DHAP. It is AP is dihydroxy acetone phosphate. So DHAP is dihydroxy acetone phosphate. Right, and the other sugar is glyceraldehyde 3-phosphate. Dihydroxyacetone phosphate here is a keto sugar, so it is a keto sugar, ketose, and it is an aldehyde. So it is in the name glyceraldehyde. So 3 carbon sugar are formed, and in each sugar there is a terminal phosphate group. You can see here that glyceraldehyde also has a phosphate attached to it, and the DHAP also has a phosphate, and these phosphates come from the 2 phosphates of the fructose 1,6 bisphosphate. So each molecule gets a single phosphate to it, and it all happens through the enzyme aldolase, which is a lyase. As I have talked earlier, lyases are for the breakdown of larger molecules into smaller molecules. Lyases only use a single substrate. As you can see here, the single substrate is fructose 1,6 bisphosphate, which breaks down by the action of lyase into two products that are the DHAP dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate. And in the next step, what happens is, because DHAP is not very stable inside the cell, so through the action of enzyme triose phosphate isomerase, it also converts into glyceraldehyde 3-phosphate. So by DHAP, by the action of enzyme triose phosphate isomerase, because it only has to form an aldehydic sugar, it is a 3 carbon sugar. So using an isomerase, because aldehydes and ketonic sugar are isomers of each other, so by using the triose phosphate isomerase enzyme, the DHAP readily converts into glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. So as you can see that one molecule of 1 fructose 1,6 bisphosphate leads to 2 glyceraldehyde 3-phosphates. So here ends the preparatory phase of glycolysis. Now the 6 carbon sugar has been broken down into its subsequent 3 carbon sugar. The conversion from dihydroxyacetone phosphate by the enzyme triose phosphate isomerase to glyceraldehyde 3-phosphate, which is an aldehydic sugar, is the 5th step of glycolysis, and this now marks the end of the preparatory phase of so at the end of the preparatory phase we have the glucose which gets converted to 2-glyceraldehyde, 3 phosphates, and in the process we have used 2 ATPs. So in the preparatory phase we have used 2 ATP molecules inside the cell to let the breakdown of a C6 carbon into a C3. Now we commence the payoff phase of glycolysis. The payoff phase where ATP and NADH will be produced. So ATP and NADH will be produced in the payoff phase. So in the sixth step of glycolysis where we have 2-glyceraldehyde, 3 phosphates, now we will talk about only a single molecule because the same steps happens to the other molecule in the cycle. So you should assume in your mind that you are using 2 reactants and 2 times making 2 products, and the other products, the byproducts that are made in the reaction are also twice in number. So now in the sixth step we have the glyceraldehyde 3-phosphate with us, and by the enzyme glyceraldehyde 3-phosphate dehydrogenase we make 1,3-bisphosphoglycerate. So here also we see that we make 1,3-bisphosphoglycerate, and the enzyme that does all the trick is the glyceraldehyde 3-phosphate, the glyceraldehyde phosphate dehydrogenase. So now we encounter a dehydrogenase, and the dehydrogenases, as we all know, are the oxidoreductases. The dehydrogenase, they are carry redox reactions, they are oxidoreductases, and this dehydrogenase forms an NADH. So here one NADH molecule is formed. Here one NADH molecule is formed, and we know that NADH is the electron donor in the electron transport chain. So NADH acts as the electron donor in electron transport chain and carries out oxidative phosphorylation. So this NADH also accounts for the further production of ATP inside the cell, inside the mitochondria of the cell, and this happens through glyceraldehyde phosphate dehydrogenase enzyme. And you might notice that we have also added a phosphate group to the 3 carbons. So here also terminal phosphate groups are added at the first and the third carbon. So what it does is that the glyceraldehyde phosphate dehydrogenase, the glyceraldehyde phosphate dehydrogenase, it acts as a dehydrogenase, but it also acts as a phosphorylase. Okay, so I have talked about phosphorylases. What they do is that they add phosphate groups through inorganic phosphate. So it adds up phosphate group to the substrate. So here we also add up a phosphate group in the reaction which makes up the whole process. So here the glyceraldehyde phosphate dehydrogenase serves 2 purposes. It acts as a dehydrogenase where it transfers the electron to the NADH and leads to the formation of NADH, and also it leads to the phosphorylation of the glyceraldehyde 3-phosphate at the terminal carbons. What it does, that it charges up the 1,3-bis phosphoglycerate, so it also gets charged up for further reactions, because it has to also undergo some breakdown in future reactions of glycolysis. So in the 7th step of glycolysis, this is the 7th step of glycolysis, we have the 1,3-bis phosphoglycerate, and it is a charged up molecule, as I have told you in the last step. By the action of enzyme phosphoglycerate kinase, it leads to the formation of 3-phosphoglycerate. So now 3-phosphoglycerate is formed, and this 3-phosphoglycerate is an acid. What happens to the phosphate group that is lost? The phosphate group that is lost is transferred to ADP for the production of ATP, and it all happens through the enzyme phosphoglycerate kinase. Phosphoglycerate kinase. So here also the kinase enzyme comes, but here it doesn't require energy. Here the substrate is already charged up, as I have talked in the last step. It is charged up. So when a kinase encounters a charged up substrate, it transfers its phosphate group to a lower energy product, that is the ADP. So here 3-phosphoglycerate is formed as a product, and the charged up 1,3-bis phosphoglycerate leads to the production of ATP. So from ADP, ATP is formed. So now it is clear why the glyceraldehyde 3-phosphate was being given another phosphate by the dehydrogenase enzyme, and why the 1,3-phosphoglycerate was charged up, because this is the first step in glycolysis where production of ATP has taken place. In the last step, where hexokinase was used, where phosphofructokinase was used, ATP was used by the cycle, but here the cycle is here because it is called as a payoff phase. So now the production of ATP is taking place. So we can also call this step, the 7th step of glycolysis, as a break-even point. Why we call it as a break-even? Because 2 ATP molecules were used in the payoff phase for the conversion of the C6 carbon into the 2 C3 products, and now the ATP molecule is produced because there are 2 glyceraldehyde 3-phosphate molecules. As I said, the cycle is repeating twice. Now 2 ATP molecules are produced in the 7th step, and that's why I am calling it as a break-even point. Now the cycle is balanced as in respect to the amount of ATP that was consumed in the preparatory phase has been given out. The output has been given out in the payoff phase, the 7th phase of the glycolysis cycle. So in the 7th step, it is a break-even point, and here also there is a kinase, but the kinase is moving in the reverse direction, because as I said that if a charged substrate is present, then the phosphate group will move from the charged substrate to make ATP, and this is what is happening, and that's why the 1,3-bis phosphoglycerate, a phosphate group, was added to make it charged, and now one phosphate has been transferred to make ATP, and the resultant product is 3-phosphoglycerate. First step in glycolysis where ATP has been produced, and the ATP production here is, as I said earlier, is through substrate level phosphorylation. No oxygen is used, only the enzyme kinase is used, and a single ATP molecule is formed. So low level of ATP always forms during substrate level phosphorylation. Now moving on to the 8th step of glycolysis. In the 8th step, the 3-phosphoglycerate gets converted to 2-phosphoglycerate. So it is just a shift of the phosphate group from the 3rd carbon to the 2nd carbon. And the enzyme that is responsible for this reaction is very simple in name. It is called as a phosphoglycerate mutase. So the enzyme responsible is phosphoglycerate mutase, and as the name suggests, mutase is an isomerase enzyme which causes structural change and no chemical change. The mutases, the isomerases, the racemases, they all come in the category of isomerase enzyme. And here also the phosphoglycerate mutase just shifts the phosphate group from the 3rd carbon to the 2nd carbon. So that's all in the 8th phase. Only a shift in structure has taken place. In the 9th step, what happens is, in the 9th step, the 2-phosphoglycerate, it gets converted to phosphoenol pyruvate. So hence there is a conversion, and it is a dehydration reaction. Water is lost in this reaction, and the result is phosphoenol pyruvate. So the product formed is phosphoenol pyruvate. It is also a 3-carbon sugar, and it is also called as PEP for short. And in this reaction, the enzyme used is an enolase. As I have talked earlier, these enzymes are lyases because they form a product that is smaller in size as compared to the bigger molecule. And the enolase enzyme, the enolase lyase enzyme, causes the removal of water. So it causes a dehydration reaction. Enolase is a lyase. It causes breakdown without the use of ATP. The thing to note is why this step is necessary is that the phosphoenol pyruvate has the highest energy phosphate in all of glycolysis. So this molecule has the highest energy phosphate. The phosphoenol pyruvate has the highest energy phosphate in all of glycolysis. So this is a very necessary step, and it leads to the formation of pyruvate in the last step, which is very essential. And the product pyruvate that is formed in glycolysis is also used up in Krebs cycle, and that further leads to ETC and oxidative phosphorylation. So in the final step, the 10th step of glycolysis, the last step of the payoff phase, because we have reached a break-even point, now the amount of ATP that was consumed in the preparatory phase has been given out till now by the glycolysis, but in the last phase, the last step of glycolysis, it will provide some energy. And in the last, as I talked earlier, that PEP or phosphoenol pyruvate has the highest energy phosphate in it. So now the PEP, which is the phosphoenol pyruvate, through the enzyme pyruvate kinase leads to the formation of the final product of glycolysis, that is pyruvate. So the final product of glycolysis is not formed, and the enzyme responsible for it is called as pyruvate kinase. Here also we encounter a kinase, and here also the production of ATP takes place, because as I have talked earlier, the substrate is high energy phosphate, so PEP has So PEP has high energy phosphate in it, so the substrate is more energetic, and when it encounters the kinase enzyme, the phosphate group has to be transferred. So the transfer of phosphate takes place from the PEP to ATP. So finally, two ATP molecules are produced because we are following up the cycle. Two glyceraldehyde 3 phosphates now leads to formation of 2 pyruvate. And also this is the third checkpoint of the cycle. So third checkpoint of the cycle, because after this point the payoff phase gets completed. Now the reaction is more on the positive side. We have more of the ATP that we had consumed earlier in the preparatory phase. We have more ATP compared to that. So this is the third checkpoint, because after that the pyruvate can enter the mitochondria, can become acetyl CoA and can enter the Krebs cycle. So this is the third checkpoint. It causes non-reversible formation from PEP to pyruvate. This is also a substrate level phosphorylation. So two substrate level phosphorylation took place, one at the time of phosphoglycerate kinase and one at this time where pyruvate kinase enzyme is used. So 4 ATP molecules are produced. So if we sum up the equation, it should be like this, that during the initial phase we had one molecule of glucose and we provided it with 2 ATPs, and what we get in result was 2 pyruvate plus 4 ATP plus 2 NADH. Okay, I have not balanced out the equation. This is the sum of glycolysis, what we get after the glycolysis gets completed. Net ATP, the final ATP is 4 minus 2 ATP, which comes out to be 2 ATPs. So this is our net ATP production after a single cycle of glycolysis. After a single breakdown of glucose, we get 2 ATP and 2 NADH, and these NADH can be used for oxidative phosphorylation as an electron donor. And this is the complete glycolysis cycle. To summarize the glycolysis, what happens is initially we have 6 carbon glucose with the enzyme hexokinase. At the expense of ATP, we form a glucose 6 phosphate, and glucose 6 phosphate is necessary because it doesn't move outside the cell, so it is retained inside the cell, and by the enzyme isomerase, the 5-phosphoglucose isomerase forms a fructose 6 phosphate, and then through phosphofructokinase we have to invest one more ATP to form fructose 1,6 bisphosphate, and this fructose 1,6 bisphosphate has terminal phosphates at the first and the sixth carbon, and it is a charged up molecule. With the help of an enzyme aldolase, which is a lyase, results in the formation of DHAP and glyceraldehyde 3 phosphate. DHAP is a keto sugar which readily converts by triose phosphate isomerase into glyceraldehyde 3 phosphate. So after the preparatory phase of glycolysis, with one glucose molecule and its breakdown, we have now 2 molecules of glyceraldehyde 3 phosphate. Glyceraldehyde 3 phosphate then under the enzyme glyceraldehyde phosphate dehydrogenase forms 1,3-bisphosphoglycerate. Here one NADH is produced, which is the electron donor that will be used in the oxidative phosphorylation to produce more ATP. And the dehydrogenase enzyme also has a phosphorylase action where it adds up a phosphate to the molecule glyceraldehyde 3 phosphates. So 1,3-bisphosphoglycerate now has 2 molecules, which means it's charged up with phosphates, and with the enzyme phosphoglycerate kinase, now the production of ATP takes place because the charged up phosphates cannot go anywhere else but they have to go to the ATP, and results in the formation of 3-phosphoglycerate. This point of the cycle is the break-even point. Output of the payoff phase is equal to that of the preparatory phase. So now the amount of ATP that was invested has been returned back to the cell. Now the 3-phosphoglycerate with the enzyme phosphoglycerate mutase, which is an isomerase, changes the position of phosphate from the third carbon to the second carbon. And with the enzyme enolase, which is a dehydrating enzyme, removes water from 2-phosphoglycerate to form phosphoenol pyruvate. And phosphoenol pyruvate is the highest energy molecule in the glycolysis cycle, so it has the highest energy phosphate in it. And with the enzyme pyruvate kinase, as I said, that the kinases, when they have high energy substrate, the 8-phosphate group transfers to form ATP, results in the formation of product pyruvate, and hence we get 2 more ATP molecules at the end. Now picturizes the detailed overview of glycolysis, and we will discuss about Krebs cycle and how ETC takes place inside mitochondria in the next video.